Okay, let's go ahead and get started. Welcome to Microbiology. I'm Dr. Van Laar, and I'm the lecturer instructor this semester. Those of you taking lab have either, I think, Dr. Tao in the morning or Dr. Kong in the afternoon for the lab, so lucky you. I had Dr. Tao for lab when I was a student. It was a lot of fun. So if you're in her class, very good. I think Dr. Kong is fun too. I just never had him as an instructor. I only had Dr. Tao. So how many of you have read the syllabus? Good. When's the first exam? 3rd of February. Very good. You did read the syllabus. Okay, if that's a surprise to you that the 3rd of February is next week and there's an exam, probably read the syllabus. We have an exam every week. Okay. Until Cesar Chavez Day, they will be on Tuesdays. So after Cesar Chavez, they will be on Thursdays. They're on the syllabus, so it's not, I'm not going to trick you and like change the dates or anything. So they're going to be in the first 10 minutes of class. They are 10 questions each. Your 2 lowest exam scores will be dropped. There are no makeups. So your 2 lowest exam scores are dropped for whatever reason. I'm sick of class. I don't want to go to school. That's fine. It's dropped. Okay, I have to go to work. That's fine. Me too. But my work is this, so I'll actually be here. If you have like military service or jury duty or something like that, and it's going to pull you from more than 2, let me know. But other than that, you don't have to tell me like, oh, I can't come to the exam because I'm sick. Like, that's okay. Feel better. Please don't come to class. We don't want to get sick. We remember what happened a few years ago and everybody would get sick, right? We don't want that. So just briefly to go over the syllabus, I'm Dr. Van Laar. My office is downstairs on the hall leading out to the door in 275. I reply to emails better than anything else. They make me put my phone number on here. Don't call me. I won't answer. It's peak millennial. Like my mom calls me, I don't answer. I'm like, no, don't call me. Text me. My mom lives down the street. I don't have to answer. I have office hours on Mondays from 9:30 to 10:30, Wednesdays from 11 to 12. If none of those times work and you desperately want to come talk to me, shoot me an email. We'll figure it out. Okay? I will respond to the email, but don't call me. When class is, because you're here, very good. The only time that class gets tricky is when we have our final. So our final is in the syllabus. I have no control. If I did, it would be different. We're on Thursday of finals week. Always Thursday of finals week. At least it's not Friday of finals week. Thursday of finals week at 11:15. So normally class starts at 12:30, but finals Thursday at 11:15. I've only had one student since I've been here be late. And I think they just forgot. Because we had this whole conversation even before the final, like the week before. Everything was fine, and then they forgot. So we're going to learn about microbes. Microbes are fun. I'm a microbiologist. I actually wasn't. I was a genetics person, and then I took this class, and I became a micro person. So if I convert one of you, I will consider my life goal for the semester to be complete. If I convert multiple, well, then I'm going to rub it in Dr. Tao's face, because I think she only got me that semester. So there is a textbook. It's mandatory-ish. You don't have to do it. You don't have to do anything. If you want the highest grade possible, you do have to purchase the textbook and do the online homework. One of the things that a lot of people ask me is, how do I study for this class? Or, well, if I'll tell you, read the textbook. It's actually really good. It's not too jargony. It's not like my OChem textbook that I would read when I couldn't sleep because it would help me fall asleep. It's actually well written. So the first thing I'll ask you is, are you reading the textbook? Because you should be. And I mean, I don't mean to sound all big brother about it, but I can see. It actually tells me if you've read the textbook. I can look you up by name and see if you've read the textbook. So if you come to me and you're like, I don't know why I'm not doing well. I'm like, you spent 1 minute looking at the textbook. That's why. It tells me how long you spent too. I guess you could trick me by just leaving it open on your computer, and I wouldn't. And then I'd be like, well, I don't know. You spent 7 hours reading it. I just can't. But the textbook is really useful. I record all the lectures and I'll put them on Canvas, so you can always revisit those. That helps to stave off the, did I miss anything important when I was gone? No, we just sat here and we waited for you. No, okay? Whatever you missed, it's there. You don't even have to depend on somebody with crappy handwriting for notes. So I'll record it and put it online. Sometimes I forget to actually put it on Canvas. So shoot me an email and be like, hey, and I'll put it up. Okay? Give me a day. Like if by the end of tomorrow, today's lecture isn't up, let me know. Don't email me in like 5 minutes because I won't be able to help you then. But it takes a little bit of time, but not that long. So expectations, please come to class. Please be prepared. Download the slides that are on Canvas. I will put up the slides as well. There's no secrets. They're not secret slides. There's nothing hidden. My slides that I'm going to present are not different from the ones on Canvas. They are the same. Please try to keep distractions to a minimum. I mean, I get it. I'm human too. I have a meeting after this from 2 to 4, and I will look at my phone. I'm not going to lie. But like this, why am I not doing well in class? Because this is how I recognize you, is the back of your phone. So if I see your phone case and recognize like your pop socket more than I recognize you, again, a problem. Okay. If anybody complains that you're doing something very distracting on your computer, I will ask you to turn it off. I had a guy one semester who just watched basketball all semester, and finally one of the other students was like, he just keeps watching all these crappy teams. I was like, okay, well, sorry he's not watching your team, but okay. So if you're watching a Lakers game, you're out! I'm a Spurs fan. I did my PhD in San Antonio. Try not to talk during class as much as possible. I can hear you. You may not think I can hear you, but in these classrooms, like it carries. And I had a couple times last semester and I felt really bad because I don't ever want to embarrass anybody, but I had to straight up be like, can you stop talking? Because I can hear you. So I don't want to have to do that. Because if it's bothering me all the way at the front, it's probably bothering your classmates. So if you want to have a conversation, you're more than welcome to step out into the hall and do so. Try to keep up as much as possible. The homework will usually be due the class period after we have finished the chapter. So whenever we finish chapter 1, I'll open up the chapter 1 homework and it'll be due by the next Tuesday or Thursday after, whenever we finish it. They are not supposed to take more than like half an hour or so. If you're really struggling and having a hard time getting through it, let me know. And maybe we can think of better like note-taking strategies and ways for you to keep up in class. Keep track. Oh, we don't really do quizzes. We just do basically the weekly exams and then the final. So just keep track. Keep track of your homework. The Canvas is pretty accurate, but you should be able to kind of determine where you're sitting in the class, like what do you need to get on the final, and then don't cheat. That's the worst too. I had to take somebody's final last semester and give them a 0, so don't do it, because like this is checking the time, this right is not checking the time, so don't do it. I've seen it. I've seen notes, I've seen cell phone in the lap, I've now seen the smartwatch. At some point, I'm assuming I'll see the glasses, whatever. Okay, I'll give you a 0. It sucks, but I will. So please don't do it. Just avoid it. If you're gonna get a C, take the C. Better than the F. Okay, there are 300 points possible in the class. 65 points come from homework. 14 exams worth 10 points each. The 2 lowest are dropped. That's 120 points from exams. In-class assignments and participation, a total of about 40 points. You have to be here for that, by the way. So again, you don't have to come to class, but if you would like an A, you have to come to class. At least a couple of times. Okay? So you can miss some classes and still do well. You can do poorly on a couple exams, you'll still do well. Don't worry about it. Okay, and then a comprehensive final. Okay, late homework will be accepted, 10% penalty per day. I'm busy, I didn't get to it. That's cool. You can still turn it in. You don't even have to talk to me about it. Just turn it in. It'll automatically give you the penalty. It'll still let you do it. It's fine. Grading scale is the standard state grading scale. I would be shocked if that was surprising to anybody. Late homework, like I said, will be accepted. Makeup exams are not given. But if you do have something like a university-related absence, like I've had athletes who had all of their matches on a exam day. Like we can figure that out. I'm not going to have you fail the class because you have matches every day, right? But otherwise, be here, no extra credit. Let's say something happens towards the end of the semester, you get really sick and you have to miss the final. That's fine. We'll give you an incomplete. You can take the final when you get better. Like, don't worry about it. Okay. Like I said, email me, please, from your university email address. If you email me from like your Gmail, half the time it gets shuffled, not even into a spam. I don't know why my Outlook has this weird setting where it's like focused and other. It gets sent to other, and I don't ever read other. I read focused. Some people get upset with me and blah, 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 blah. So just use your stand state. You are not allowed to record the class unless you have specific accommodations from DRS. But I record the class as well, but those recordings are for the use of students in this class only. If I feel like they're being shared outside of class, then I will stop recording the class for everybody. So don't ruin it. Okay. Attendance, it's your choice whether or not you come to class. If you choose to not attend, you would forfeit any points that might have been earned that day. And I think that's just kind of the risk, right? You don't go to work, you don't get paid, you don't come to class, you don't get the credit. It is what it is. Please don't cheat. I hate that so much. There is tutoring available through Stan Tutor. They just sent me an email literally right before class. What did they say? They said tutoring begins Tuesday, February 10th. Appointment scheduling opens Wednesday, February 4th. So there's both scheduled and drop-in tutoring. Drop-in tutoring will be downstairs on the first floor Monday through Thursday from 4 to 7. So there are options to seek help either from me or from the Stan Tutor as well. Okay, Writing Center, DRS. If you feel like you are going to want extra time on exams or you need an accommodation to help you be as successful as possible in the class, please pursue that. I can't give an accommodation unless I get a letter saying that the student has been approved for an accommodation. And do please take advantage of it. Okay, if you've got it, take it. If you think you might need it, go ahead and contact DRS and they can be really helpful with that. Food insecurity, definitely a real problem. If you need food, please visit the food pantry, student health center, and cross-cultural center, undocumented student services. Important dates, last day to add or drop classes is February 23rd. If you make it to the 23rd, you might as well just stick it out the whole time. I feel like that's really late, but okay. Cesar Chavez Day is March 31st. That's a Tuesday. Yay. We don't have class. I don't know why it says the 26th. That's weird. It's the 6th through the 10th. Basically, right after Cesar Chavez Day, we have that whole week off. You can come, but I won't be here. Last day of classes is May 15th. I think for us it's the 14th because 15th is a Friday, and then our final is on the 21st. I don't know why this says lecture online either. Those are in-person lectures. That's fine. But the exam schedule is correct. Sometimes we can stick to this lecture schedule. Sometimes we don't, but that's okay. The most important thing here is that you know when the exams are. And I try to make it easy. They are every Tuesday until right after Cesar Chavez when it switches to Thursday. And then they will be every Thursday until the end. Okay. And then here's our no class days. So the only holidays we get this term are Cesar Chavez and spring break. Okay, questions, comments, concerns about the syllabus? Okay. You may have noticed if you did log into Canvas already, most of you did, that right, there's recommended readings. It is not mandatory that you read. You'll see it pop up as an assignment only because I'm just telling you this is the chapter that we're going to be covering. This is the information I really recommend that you read, but it's worth 0 points. So it's totally your choice if you want to read and engage that way. When the homeworks become available, I'll have a module that says homework, but it will also show up on your to-do list that you'll have a homework assignment. And you'll know that there will be a homework assignment coming if we finish a chapter. And again, if I forget, that's my bad. I would extend it out. I would extend out the deadline. So if I forget to assign it until a few days later, like let's say we finish chapter 1 on Thursday and I forget to post the homework until Saturday, I wake up and I'm like, oh man, I would extend it, obviously. I'll give you until that class session after. Okay, and then you access Pearson through here. If you have any issues with Pearson, I can't help you. I always have issues with Pearson too, and I call them all the time. I can give you the lady's number. In issues with Canvas, I can kind of help you kind of figure out what's going on, get you at least over to OAT. Issues with Pearson is a Pearson thing, but it usually, at least for me, right, it looks like this. Then I can open it up. And like I said, the textbook is actually really good, Pearson eText. Oh, obviously I was reading chapter 27 last time I opened this book. So, okay. There we go. Start from, there's my outline, here we go. Chapter 1, the microbial world in you, right? It gives you learning objectives. And again, it's really straightforward to read. They have these really nice clinical cases for those of you interested in like clinical microbiology. And it again is not written to be like for people who already have a PhD in microbiology. It's written for students who are learning microbiology. So I really like it. And the chapters aren't terribly long. They won't take, too long to read. So that's your book. You're already paying for it. You might as well use it because you have to pay for it to do the homework. So I hope you'll use it. But again, that is 100% your choice. Any questions? And you also pay for the gym. It's your choice if you want to go to the gym. You pay for a lot of stuff as students. I hope you take advantage of all of it. I'm like, you're taking my money. I'm going to take everything you're going to give me. Okay, so let's go ahead and start with chapter 1. Anytime you have questions or you want me to clarify something, please feel free to raise your hand. If you don't think I've seen you because you've had your hand up for a really long time and I haven't looked at you, please feel free to just say, hey, excuse me. And I want to make sure that questions get answered like when they are coming up rather than hoping that you remember it by the end of class. And then on some Tuesdays, like today, I have to leave because I have a meeting across campus at 2, so I won't be able to stay very long after. So if you have a question, ask it right now. If you have a question, probably other people do too. So don't ever hesitate to ask questions. Yes?
So for the Pearson thing, is there an access code for the class, or do you just have to pay for it and then register?
I think you should pay for it and then register. Yeah, there's no specific access code. You should be able to just click on the link on Canvas and go through. If you have any trouble, I'll give you those contact information. And especially if like multiple people are having trouble, I can always extend homework deadlines until we kind of get it all figured out. It's not a big deal. Yeah, I'm not going to penalize you because some weird tech system isn't working. Okay, cool. So, what is microbiology? What are microorganisms and what do they do? And that's basically what we're going to cover for the whole semester. Microbiology, if you're a word nerd, the study of small living things. Microbiology, study of small living things. And by small, I mean microscopic. You have to have a microscope in order to actually visualize the individual microbes, with a couple of exceptions. As you'll see later in the semester, or if you kind of look in a little bit more detail at the topics we're covering in the syllabus, we do cover eukaryotes, like fungi, which can include mushrooms. And you would probably think about, well, I can see a mushroom without a microscope. Yes, that's true. That's true. But there are many unicellular fungi that you can't see without a microscope. So there's my caveat there. We also cover parasites, things like tapeworms and roundworms. That's actually one of my favorite lectures. That's a fun one. You can also see those, unfortunately, with your naked eye. But the larval stages are microscopic. So I guess we got a little greedy as microbiologists. We're like, we want these too. So we brought all of those in with us. So those are kind of from bacteria and viruses to some of our multicellular worms. That's what we're going to cover in this class. And those all really fall under that umbrella of a microbe. Now you'll notice there's a parenthetical statement here. It says, includes non-cellular agents. And we'll get a lot more into this as we get into the semester. But you may have covered this concept in other classes. I've actually never taught the Biology 1050, so I don't know how in-depth you get into that class. But viruses don't have a cell. But they are considered microbes. And we cover them in this class. So that's what we mean by non-cellular agents. Viruses, viroids, and prions will be falling under this umbrella. And then, of course, what microorganisms do. I imagine a lot of you chose this class, right? It's not required for the major. A lot of you chose this class as one of your diversity and systematics classes, for those of you who are bio majors, because you are in some way interested in health. Is that relatively accurate? How many of you are interested in some sort of health profession? Yeah, that makes sense. You might be interested in, pharmacy or medicine or veterinary medicine, optometry, whatever. You want to work with something and help either people or animals. And one of the major things that you have to contend with as a healthcare provider are infections. Infections kill people all the time. I did my postdoc with the U.S. Army, their Institute for Surgical Research, in the Dental and Trauma Research Detachment. And this was, of course, when we were in combat in Iraq and Afghanistan. I was not in the military. I mean, look at me. I was not in the military. I was a civilian doing my postdoc with the military, specifically the Army. And but we did talk a lot about battlefield medicine, and battlefield medicine has gotten to the point where 90% of soldiers wounded in combat will survive the initial wound. And when I say wound, I mean something as complex as having all of their limbs blown off. Burns over 75% or more of their bodies. They will survive that. There are actually very few non-survivable wounds at this point. And then they get an infection and die. And that's terrible. Could you imagine? Like, they've survived all of that, and then they come home and die of an infection that they got in the hospital from y'all or from the other patients, right? Because when you think about a situation like that, we always use the example of Iraq. They were at some sort of like forward operating facility. They were injured. They were usually evacuated to Baghdad. Then they were evacuated to Germany. Then they were evacuated to sometimes Walter Reed, and sometimes where we were, I was in San Antonio at the Brooke Army Medical Center, or now it's called the San Antonio Military Medical Center. And it could take 7 to 10 days. The goal there was, please don't let this person die. So during that whole transport, they're like, please don't let this person die. Not infection control. It was just survival. And unfortunately, they would get infected somewhere along that route and succumb to those infections because they were already weak. Right? And a lot of times those infections are antibiotic resistant, which is another thing we'll talk about. So, very long story to get to the point that I understand that the majority of you are taking this class because you are interested in things like that. How do you keep your patients from dying? How do you protect yourself when your patients have a scary infection? Right? When the nurses went down with Ebola in Dallas, You don't want to be that one. They survived, but that's a risk. Healthcare providers who died of COVID, right? You don't want to be that person. So you're interested in infectious diseases. But microbes have so many more important roles in the ecosystem that I hope those of you who maybe aren't interested in human health and are taking this for some other reason as a diversity and systematics class will also find something really interesting. Their roles in ecosystem foundation, ecosystem shaping, part of our normal microbiome. So they are hugely important, foundational to life on Earth. If this Earth had no people, the Earth would be fine. It would actually probably be better because we mess it up. If this Earth had no microbes, we wouldn't last very long. So, they're really foundational. And we'll spend time talking about all of these different aspects. And again, that popular view is that microbes, well, a lot of times the popular view is that microbes are bad. They cause infection. They're bad. And yeah, a lot of time they do. And they impact us most significantly, in our own opinion, when they are causing an infection. I'm sick. You have a wound. We call them germs, right? They're bad. That's what my kids say. They know germs. My kids are 4, so they know about germs. Germ just means a rapidly growing cell, which is pretty accurate for a lot of microbes. And so we'll talk a lot about microbes and human welfare, not just in the aspect of like infections and medicine, but also in how they support us to be as healthy as possible, how we can adjust our microbes to try to be healthy, and how important they are in the ecosystem. So microbes are super important to human welfare. Why do we study them? Because they're awesome. That's why. I think microbes are really cool. Microbiology, I'm not just saying that because you're in here. It's my favorite subject to teach. Okay. Some of you were in my class this morning and I did not say that it's my favorite subject. Just saying. I love it. Don't get me wrong. Okay. But this is my favorite subject. I am a microbiologist through and through. I love microbiology. And I hope that even if you don't become a microbiologist, you'll have an appreciation for the subject because microbes are everywhere. We are encountering them all of the time. As you are breathing, and I know you're all doing that because you're all still awake. As you are breathing, you are inhaling microbes and you're exhaling microbes. And those of you who are close to each other are inhaling and exhaling each other's microbes. I'm a little bit farther away, so I don't have to do that. But I'm drinking coffee. It has milk in it. So there's some microbes in there. You ate food. It has microbes on it. You drink water. It has microbes on it. Everywhere has microbes. And they have, I can't overstate the impact they have on humans. And again, we often focus on the bad, but the good cannot be overstated. The positive benefits that microbes have on people are incredible. So why do we study them? Well, one, because they're interesting. Especially if you're a biology major, hopefully you're at least interested in living things. Studying life, might as well notice the world around you, be curious. So study them because they're there. That's fun. But we can also study them to improve our lives. How do we prevent our food from going bad? I told you my kids are 4. They basically live on fruit. Do you know how expensive fruit is? Do you know how mad I get when I open the refrigerator and the fruit that we just bought is already spoiled? Right? Big sigh. Or you spend a lot of money on meat, or remember when eggs were super expensive? Okay. How do you, if you spend your money and your food goes bad, you're either going to eat it and get sick, or you're going to have to throw it away. So we really want to prevent food from spoiling. And that's not to mention the microbes that we eat on a daily basis on purpose. Mushrooms on your pizza, anybody? Bread, beer, wine, kombucha. We can use microbes to prevent or treat disease. By understanding how diseases are transmitted, we can break the cycle of transmission. When it turned out that all of these weird diseases are transmitted by mosquitoes, we're like, well, let's get rid of mosquitoes. That helps a lot. Hey, we're building the Panama Canal and everybody's dying of yellow fever. Let's try to get rid of the mosquitoes. We also use microbes to discover antimicrobial agents. Antibiotics, not the first antibiotics, but a lot of antibiotics, some of the early antibiotics, were discovered because they were produced by other microbes as a type of competition. You're going to take my space, I'm going to make this thing that kills you. So when we look to the microbes, we can see how they've evolved over millions and millions of years together to have that sort of biochemical warfare, and we can take advantage of it. And we know that microbes can cause disease. They've killed more people than all wars. More people died of the flu after World War I than died fighting in World War I. A lot of people died fighting in World War I. In the US, it's estimated that there are 750 million cases of infectious disease every year. Do you know how many people live in the US? Like 300 and something million. So y'all getting sick at least twice, is basically what that's trying to say. And again, it's estimated that nearly 250,000 people every year will die of an infectious disease. Do you know what's going around right now that's killing people? The flu. Young, healthy people are dying of the flu. I got my flu shot. I recommend you get yours because you are young, healthy people. And it costs tens of billions of dollars. And you don't have to answer this, but think about, do you have health insurance? Do you have good health insurance? Do you have a job that will pay you to not be at work? Can you pay your rent if you can't go to work? It's expensive to be sick. Okay. And then we talked about food spoilage, like if your, meat rots or your vegetables and fruits rot, your milk gets all chunky. Milk is not supposed to be chunky, right? You lose all that money. Again, it's expensive to throw away food. But also foodborne diseases. How many of you like to go out to eat? Like at a restaurant or, anywhere. Not even a fancy restaurant. You're like Taco Bell. Like, how many of you like to go to Taco Bell or McDonald's or whatever, In and Out? I hope I don't get diarrhea, right? Is that the thought you have when you walk in the door? Well, if it is, you're probably a microbiologist. But you trust that the food products you buy are not going to make you sick. I trust that if my kids want to go to the fry store, which is In and Out, that they'll be fine. That's not always something you can trust. People of my age, we don't go to Jack in the Box. We don't do it. Why? Because our mothers would have killed us if we'd gone to Jack in the Box. Why? Because kids died of E. coli they got at Jack in the Box when we were kids. You buy your kid a burger and they die. Like, that is not supposed to happen. So it's really important that we also can protect foodborne diseases. And you guys may not remember this, but there was an outbreak of norovirus that hit a bunch of Chipotles. This Chipotle too. I lived in San Antonio at the time, and I was like, wait a minute. I've been to that Chipotle. My dad likes Chipotle. I've been to that Chipotle. I don't like that Chipotle anymore.
I have a question out of curiosity. You know how you said food goes bad really fast? Say if you got strawberries and you have molds on 3 of them at the bottom, do you have to dispose the whole entire carton?
Oh, we don't. We eat them. If they don't have the mold, if they're not rotten, like if they're not like soft and squishy, we'll still eat them. They're usually going to be okay. If they have visible mold growth, no.
I would assume like they have mold on them, you just can't see it.
Right, and it's usually not enough that it would make you sick. I hate to, there's,
Because my dad tells me like you can still eat them,
Yeah.
it's fine, but I think that's gross.
it can be, but if you've ever gotten sick from eating anything, I can tell you right now you've eaten something that had somebody's poop on it. Whether or not it was an animal or a person, it was poop. And you eat poop bacteria every day. You eat and drink it every day. If you drink milk, there is a USDA approved level of fecal contamination in your milk. They call them coliforms, bacteria that are associated with fecal contamination. So there are, there are acceptable levels of contamination, and it's fine that it gives you the ick. Like, that's totally okay. But the amount is usually going to be so low that your stomach acid would be able to tolerate it. Plus the fruits are kind of acidic as well. But when it has that visible growth, really what you're looking at with mold is not even that the molds are going to make you sick. It's that the molds produce toxins that cause intoxication that can kill you. Yeah, so some of the molds produce like ergot, which is like an LSD. LSD is a derivative of ergot. Other molds like Aspergillus produces one called aflatoxin, which is the most potent natural carcinogen. And I don't know if you knew this, but at least 3 people have died in Northern California from foraging mushrooms. So this like within the past 2 months. So mold toxins are very dangerous. Mold consumption usually is not. It's just more of a, well, now it doesn't taste good and it's squishy and weird looking. But I'm also the person that's like, well, it has a little weird spot, so I'm gonna like cut off 90% of the strawberry. My kids don't care, but I care. So I do cut off the little squishy parts. In theory, it shouldn't make you sick. But yeah, toxins are what I would worry about with things that had mold contamination more than anything. Mm-hmm. Yeah. Don't go out into the forest and pick mushrooms. Like PSA, if you learn anything from this class, if this is the only thing you take home, do not go out and pick and eat mushrooms. Because you'll die. 3 people have died. At least 2 or 3 need liver transplants. One's a little kid, has to have a liver transplant. So don't eat mushrooms. Unless you buy them at the grocery store. Don't trust anybody. I actually don't trust anybody. I don't like to go to people's houses and eat food that they cook. I don't trust them. Okay. So microbes are, again, commonly associated with foodborne illness. I will never eat undercooked beef, ever. Ever. Disgusting. Gross. Enjoy your tapeworms. Have fun. Yeah. Never, ever, ever. I felt bad because we were in Boston over the summer and we went to this restaurant and they had these, like, I don't know, $20 hamburgers. I'm like, oh my God, why is everything so expensive? Were you in Boston, New York City? I don't remember. It was a $20 hamburger and I was so mad. And then it was undercooked and I was like, oh, I can't even eat it. So I had to make them take it back and give it me a hockey puck, please, so I won't get sick. But, overall, microbes are actually really helpful. There are comparatively few pathogens when you're considering microbes as a whole. Even, thinking about like viruses. There aren't that many viruses that infect people, maybe other animals or other systems, but okay. So there are a few bacterial species that actually cause disease in people. And we are completely covered inside and out with bacteria. There are very few parts of our body that are sterile. We do run into problems when those parts become unsterile. So your blood should be sterile. Your lungs, like deep in your lungs, should be sterile. Your pleural and peritoneal cavities should be sterile. Otherwise, all bets are off. You got bacteria. And fungi. Bacterial viruses, pretty common in there too. Okay. And we call that our microbiome. It's normal. It is okay. A lot of times people will consume food products or supplements to help balance the microbiome. The microbiome is well studied in terms of its effects on the gut health. Having a dysregulated gut microbiome can lead to gut health issues. A healthy microbiome help you have a healthy immune system. Your immune system is constantly surveilling and sampling, and so it learns from your microbiome. So there's kind of this steady state of your immune system being primed and ready and right there in case something bad happens. The microbiome has also been, I don't want to say associated, I'll say correlated, because we know correlation does not equal causation. And I'll give you an example. Violent crime and ice cream sales increase at the same time. Do violent criminals like to buy a lot of ice cream? Does ice cream make people violent? Probably neither of those things. Turns out when it's hot, people get grouchy and also want ice cream. So correlation is not always causation, but we do tend to see there are certain microbes that are present at higher levels in humans and other animals with obesity, and are at lower levels in humans and other animals that are lean. So the question, when I say correlation does not equal causation, do those microbes cause obesity or leanness? Or they just happen to have them, right? There's some other effect. Or obesity causes these microbes to be present more than others. We don't know, but they are associated, correlated. And a lot of people are looking into what we call the gut-brain axis, right? Every part of your body talks to every other part of your body. Do the microbes have an effect on your mental health? And I leave that as an open-ended question. I don't know the answer, but it is an area of active research. Okay, so I told you microbes are really important. Of course, you understand how important they are for human health, but microbes generate more than half of the oxygen we breathe, excavate huge underground caverns, contribute to changes in our climate, make up the largest mass of living things on Earth. You can't even see them. Life originated with microbes. All of life is derived from microbes. Life, and I think I said this earlier, without higher organisms, like us, is possible. Life without microbes is not. So, I mean, this is from the American Academy of Microbiology, so, you know, maybe not a non-biased source, but it is hard to argue with that fact because we know it to be true. Okay, so microbes are important in recycling vital elements. They are critically important things like decomposers in the ecosystem to recycle carbon, nitrogen, sulfur, phosphorus. Microbes are the only organisms we know of. There are only a handful of organisms that we know of, and they're all bacteria or bacterially derived that can take nitrogen out of the atmosphere and turn it into a bioavailable form of nitrogen. Call it nitrogen fixation. Microbes can do it. Nothing else can. And nitrogen is really the limiting nutrient for things like plants. So nitrogen is so important that this is the root system of a plant in the legume family, peas, beans, clover, that they will form these associations where the root will actually recruit bacteria to cause an infection because the bacteria will produce nitrogen for the plant. They need it that bad. Primary producers, microbes do a ton of photosynthesis. Decomposers of organic waste, we take advantage of microbes for our water treatment facilities. So sewer treatment and water recycling takes advantage of using microbes to clean the water, not chemicals. Okay, and bioremediation. This is a huge area of growth, to use microbes to degrade things that we call xenobiotics, so chemicals that never existed before. They've never been seen before because we made them, because that's what people do. They can degrade hydrocarbons, like oil, and they can turn heavy metals from toxic to less toxic forms. So they're really important in cleaning up our ecosystem. We can also use them as biological insecticides. Many plants in the United States are what we call GMOs, genetically modified organisms. One mechanism or one type of genetic modification is it introduces a toxin gene. The toxin is called Bt toxin. It's from Bacillus thuringiensis, and it's toxic to herbivorous insects. So when they eat it, they die. It is not toxic to people or other like animals, like vertebrates. It's toxic to lepidopterans and caterpillars, right? Things that will, the larval forms of insects that will consume plants. And it's an alternative to something like DDT, which is a chemical pesticide. Carcinogen, right? Basically cause bald eagles to almost go extinct. So we use them in that way. We also use them in biotechnology and recombinant DNA technology. We use microbes to enhance production of various chemicals. Of course, if you are interested in fermented beverages, beer and wine are classic examples where we use microbes to produce ethanol. That's what makes beer and wine instead of, grape juice. Acetone, acetic acid, lots of other like industrial chemicals. Biofuels, using microbes to break down, I know they don't like to call it bio-waste anymore. I forget what they like to call it. That's okay. Bio-waste, like agricultural waste products, break them down and produce ethanol that can be used as a fuel for the cars. And we'll talk a lot more about other types of manufactured products. If any of you are type 1 diabetic or know someone who is, insulin is produced by microbes. If you've ever had a Hep B vaccine, it's produced by yeast, okay, microbes. So chemicals, proteins, drugs, right? Production of crops resistant to pests, drought, or temperature. Gene therapy for genetic diseases. How many of you have heard of CRISPR? We found CRISPR in bacteria. It's their immune system. It's how they fight viral infection. But now we're trying to use it to, fix human conditions. Obviously in food production, one of my favorites, cheese. Okay, cheese, bread, coffee, tofu, sauerkraut, whatever. So not only can we use the metabolic capabilities of these organisms to make our food. Why does your bread rise? Your sourdough bread, right? It rises because the yeast are producing carbon dioxide. Why does beer have alcohol and bubbles? Because yeast produce ethanol and carbon dioxide. Saccharomyces cerevisiae. If you speak Spanish, cerveza, cerevisiae, that's what it is. Brewers or baker's yeast, depending on what you're doing with it, brewing or baking. Maybe not the same thing, right, at the same time, but same species. Okay. So we can either use their metabolic processes or we can consume them directly as food. Spirulina is a cyanobacteria. Seaweed, right, is algae. So if you like sushi, and then of course, like, mushrooms on your pizza or in your salad or wherever you like to eat mushrooms, but do not pick them from the forest, okay? The ones you bought at the grocery store. Questions to this point? If you can't tell that I like microbes, well, we're going to have a fun semester. I love microbes. Okay. Another thing that scientists like to do is classify things. We like to figure out what things are, give them names, talk about them. And so you've probably covered this in other biology classes, but we have a scientific standardized naming system. We call it our binomial nomenclature. So every species has basically two words that make up the complete species name. Binomial, right? Two names that make up the complete species name. They are Latinized. They're not real Latin words, but they are Latinized. So you could take any word and make it sound Latin-y, and you can use that as a species name. Like people have named things after Lady Gaga or Barack Obama. They named a new spider species after our dean. So like you could take anybody's name and turn it into Latin-y. His name was not Carolus Linnaeus. That's his Latin-y name. His name was Carol. So we have the genus, which is always capitalized, like the first letter is always capitalized. We have the specific epithet, which is the second name, and it's always lowercase. But you have to say the two together. And the reason you have to say the two together is because a lot of times these names can be recycled, like an individual name could be recycled. If I said that I'm talking about the species coli, do you have any idea what I'm talking about? You might think I'm talking about E. coli. What if I'm talking about Balantidium coli or Acanthamoeba coli? Very different. Vulgaris. Everything is vulgaris because it just means common. So vulgaris what? Proteus vulgaris? That's a bacterium. Sturnus vulgaris? That's a bird. So you have to use both. So why can't you just use the first name? Okay, Staphylococcus. You might go, well, yeah, Staph aureus. Okay, but what about Staph epidermidis? Or Staphylococcus saprophyticus? There's a big difference between Streptococcus pyogenes and Streptococcus agalactiae. So you just have to use both names. When you write them down or type them up, well, usually when you type them, you would italicize them. So if you're ever writing something, you'll italicize, like if you're writing a paper on your computer. But if you're actually handwriting, like technically you're supposed to underline. That's to indicate that it's the Latinized form. So they are Latinized. They are used worldwide. Common names can be very different. The word for dog is different in basically every language. Right? Some languages that are sort of related, like the Romance languages, may have similar words where they can kind of figure it out. And sometimes you're like, I don't know what that means. So if I'm talking about a cow and somebody's talking about a vaca, we think we're talking about different things. That we're actually talking about the same thing. But if I knew the Latin name for cow, which off the top of my head I don't, we could be sure that we were talking about the same thing. So it allows people who speak different first languages to have that same scientific language in common. The names themselves can derive from a lot of different things. They can describe the organism, maybe the way it looks, its shape, where it's found. They can honor a scientist. So I told you there's a spider species that they named after the dean. Or they could honor somebody else, like Lady Gaga. Not a scientist. She's awesome, but she's not a scientist. They could identify the habitat. So like coli, colon. Treponema denticola, found in your mouth. Porphyrimus gingivalis, found in your mouth. So it really depends. So Escherichia coli, both is named after a person and a place. The scientist, his last name was Escher, and it's found in the colon. So the genus honors a scientist. The specific epithet tells you where you find it. Staphylococcus aureus, this is a more descriptive name. So Staphylococcus, so staph means like group, cluster. Coccus means circle, so it's a circle bacteria that grows in a cluster. And aureus is Latin for gold. So it's a yellow bacterium. And of course you can abbreviate it. Most people don't say Escherichia coli. I think I'd been a microbiologist for about 10 years before I felt actually comfortable saying Escherichia. I still don't know if I could spell it really fast. I'd have to like think about it before I spelled it correctly. So we say E. coli. Staphylococcus aureus, the scientific way to abbreviate it would be S. aureus, but you could say, most people say staph aureus. So you can shorten them, but only once everybody knows what we're talking about. So if I'm writing a paper about E. coli, I have to at least once spell out Escherichia coli, and then I can say E. coli in the rest of my paper. Okay, your first quiz. Which scientific name or names, there may be more than one correct answer, are correct? You can talk to each other, bounce an idea off one another, and then we'll do an informal poll. So we'll spend 30 seconds. You can talk to each other. Please do talk to each other. Talk to each other, and then we'll come back and go over the answers. Which is correct of a scientific name? Okay. And again, this is obviously not, I don't know your names, and I'm obviously not keeping a record of who gets an answer right or wrong, so don't feel bad if you get an answer wrong. Don't be shy. Baker's yeast, is that a correct scientific name? Oh, very good. Very good. That's why I let you talk to each other too, then you feel a little more confident. How about the way B is written? Saccharomyces cerevisiae.
No.
Why not?
Not italicized.
Not italicized. Okay, how about C?
Yes.
Good or bad? Good? Yes, because it's italicized. How about D? Yes.
Yes.
Right? Underline or italicized. So C and D would be correct here. Cool. See? Good job. Okie dokie. Continuing on. And of course, if there are any questions, but I feel like a lot of this is probably review for most of you, but if I'm ever going too fast, just let me know. Broadly, we classify cellular organisms. I'm not talking about viruses right now. Cellular organisms are classified because they have one of two cellular morphologies. They either have a nucleus or they don't. That's the big one. So we have prokaryotes, pro meaning before the nucleus. Eukaryotes, true nucleus. Of course, like the big difference is the nucleus. We will get into more detail about differences between prokaryotes and eukaryotes and why a lot of microbiologists don't even like the word prokaryote, but we still use it anyway because it's the best one we have. Mostly this class focuses on organisms that are prokaryotes, but again, we will touch on and spend some time with some of the eukaryotes. And then of course those weirdos without a cell. So, just to kind of briefly introduce some of the different kinds of microorganisms. And again, I'm anticipating that a lot of this is review. So hopefully the first week or so, you're like, okay, it's been 6 weeks since I've been in class, kind of get the brain working again before we start diving into different stuff. So first we'll talk about bacteria. So bacteria are a whole domain of life. Right? There are 3 domains. Bacteria are one of them. They are prokaryotes. They lack a nucleus, All bacteria lack a nucleus. If you have a nucleus, you can't be a bacterium. You can't play. Sorry. Got to be in your own group. They're unicellular, for the most part. Sometimes they'll come together and form these weird, like, multicellular slime molds, but that's okay. Most of them have a cell wall, and the cell wall is made of something called peptidoglycan. And we will talk more about peptidoglycan, I think in chapter 4, when we talk about the cell wall. But the differences in the way that bacteria put together their peptidoglycan, for those of you interested in clinical medicine, has a lot of important implications in the clinical setting. They don't do mitosis. They don't have a nucleus. Mitosis, by its definition, implies nuclear division. They divide mostly by binary fission. They just kind of split in half. Some of them can move. Most of the ones that move do so through a structure called a flagellum. Some eukaryotic cells also have a flagellum. The sperm cell is the classic example of a eukaryotic cell with a flagellum. It's the wavy tail at the end. A bacterial flagella are made of a totally different protein, and they move in a different way. Bacterial flagella move using a rotational motor, almost like if you picture a propeller on a boat, how it is in a circle, it moves in a circle. That's how their flagella move. There's literally a set of motor proteins that cause it to rotate. And if it rotates one way, it does one thing. If it rotates the other way, it does another thing. Eukaryotic flagella typically beat, like a waving motion to swim. So they're made of different proteins, and they move in a different way. But we call them the same thing, because why would we make it simple and call them different things? Microbes, bacteria specifically, can get their energy from a number of different sources. Some of them are photosynthetic, so they do photosynthesis. They can use light energy to fix carbon dioxide, like to take carbon dioxide and turn it into carbohydrates, and then use those carbohydrates for energy. Some of them use organic molecules for energy. That's what we do. We break down carbohydrates, lipids to generate energy. Some of them can use inorganic chemicals to generate their energy. Hydrogen sulfide, hydrogen, nitrate. It depends on the microorganism. Of course, some are pathogenic. And again, I think that's probably what most of you are interested in. And don't worry, we will talk a lot about those kind. The field of studying bacteria is bacteriology. Now, this class used to be called bacteriology, but we talk about more than just bacteria, so we changed it to general microbiology. There's a whole other domain of unicellular organisms that lack a nucleus. They're called the Archaea. So I used to know an invertebrate zoologist at Fresno State who would get all uppity about, oh, bacteria, who cares, blah, 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 yeah, eukaryotes. And I'm like, dude, we're 2 of the 3 domains of life. You study crabs. Like, come on. Like, he said actual, little, crabs, and he only studied a couple species. I'm like, come on. Like, so you can get the biologist to, get a little scuffle. So we're 2 of the 3 domains of life when we talk about prokaryotes. Archaea are kind of weird. They're often what we would call the extremophiles. You typically find them in maybe very acidic or very alkaline conditions, very high temperatures or very low temperatures. These are classically organisms that we have found living in areas that we thought were inhospitable to living systems. They most of them will have a cell wall. It is a different cell wall from that bacteria have. It's made of a different component. And again, we'll talk in more detail. I believe it's chapter 4 about how the cell wall is structured. And I gave you, the example, they can live in highly acidic or, high temperatures. So they can be thermophiles. They like really high temperatures. They can be extreme halophiles. They can live in very salty conditions like the Dead Sea. Not so dead after all. Microbes can live in there. Some of those really high salt areas that are pink, they're pink because of the bacteria or the archaea that live in there. The archaea produce a pink pigment. They can be methanogens, so that means they generate methane. Methane is actually a more potent greenhouse gas than carbon dioxide. And one of the biggest contributors to greenhouse gases, the methane greenhouse gases, are the archaea that live in the guts of cattle that help them digest the plant material they eat. And so cattle release the methane produced by these archaea, and that is a very potent greenhouse gas, mostly through burping, but sometimes through the other way. As far as we know, there are no archaeal pathogens, as far as we know. And that's good, because some of our best antibiotics work really well against peptidoglycan. They don't have peptidoglycan, so the antibiotics wouldn't work. So it's good that they don't cause pathogens because we lose a whole subset of our drugs. Now we can talk about some of the eukaryotes if we have to. No, I think eukaryotes are also really interesting. They play really critical roles in the ecosystem, especially fungi and algae, really foundational to the ecosystem ecology. Fungi also have a cell wall. It's typically made of chitin, which is a different carbohydrate. They're all carbohydrate derivatives: peptidoglycan, pseudomurin, chitin. It's all carbohydrates, just how they put them together. Again, as far as we know, there are no photosynthetic fungi. They all require organic chemicals to get energy. And that's why we typically find them as what we call saprobes. They are decomposers in the ecosystem. When fungi are in a unicellular form, we call them yeasts. When they are multicellular, they are molds. If they're fleshy, we call them mushrooms. Some fungi, especially pathogenic fungi, are capable of transitioning between a mold and a yeast form. The ones that can do that are usually very, very dangerous. We have one such pathogen in the valley. It causes a disease called valley fever. We're very creative. Sometimes they call it San Joaquin Valley fever, but valley fever is caused by Coccidioides immitis. It is a fungus that is capable of doing that. Very, very dangerous infection. The field of studying fungi is mycology. We also have our unicellular eukaryotes that aren't fungi or algae. So we call them protozoans. They're protozoans. The taxonomy of some of these organisms is really challenging. With things like DNA sequencing and more detailed computational analysis, all of the phyla have changed. Like the phyla that existed when I was a student don't exist anymore, and there are all of these weird different classifications. Even with the bacteria, they renamed all the bacterial phyla. So you'll find there's a couple chapters in the textbook about like classifying microorganisms like taxonomically, and I can't even teach it because it is too old. This is a brand new textbook, it's too old. Most protozoans also will absorb or ingest organic chemicals. Some of them can be motile, and they can move through different mechanisms. Some of them will move through flagella, but remember these are eukaryotes, so their flagellum is a little bit different from that of bacteria. Some of them will use cilia. If you've ever looked at things like Euglena under the microscope, those are protozoans that use cilia. We have cilia too in some of our cells. Some of them will also move through what are called pseudopods. These are like extensions of the membrane. They kind of pull make it sound really creepy, right? You pull out your membrane, like drag yourself along. False foot, that's what it means. The amoebas do this. They're real common to have pseudopods, to make like the pseudopods. They can be free living or parasitic. Sometimes they can do both. They can be free living and then get into a host and cause really significant disease. The brain-eating amoeba, Naegleria fowleri, is a real classic example there. But sometimes they are pure parasites. They have to go from host to host to host. It just really depends. Some of them can be photosynthetic, like the Euglena, right? The ones that are green usually are photosynthetic. The field that studies protozoan is called protozoology. I actually don't know anybody who studies just straight up protozoans. Mostly they're parasitologists who study that. We can also have unicellular or multicellular algae. They have a cell wall as well. It's made of cellulose, different carbohydrate. They can be found in different kinds of water, fresh, salt. They can even be found in soil. They are photosynthetic. Typically, when we think of something photosynthetic, we always think of green. And I told you earlier that like the seaweed and kelp, they're all types of algae, and it's not green. Depending on where they are in the water column, there are different pigments that allow photosynthesis. So the ones that have to use the green pigments tend to be near the top. If they can use different, or the ones that have the green pigment, they tend to be near the top. If they have brown or red pigments, meaning they can use different wavelengths of light that penetrate deeper, they can extend deeper in the water column. So that's why things like kelp can be very, very long, because they can use different pigments. Algae have a lot of important applications, especially nowadays for like development of things like biofuels and biotechnology. In terms of human pathogenesis, we typically worry about algae that produce neurotoxins. They can produce very potent neurotoxins and cause like red tides. The study of algae is called phycology. If any of you know Dr. Arun, he studies algae, particularly for its biotechnology applications. Really quickly, viruses, and we'll have a few chapters on those. They don't have a cell. They either will have RNA or DNA. They have one type of genetic material, not both, and they don't switch. They will have a protein coat that surrounds their genetic material. Some of them may or may not have a lipid envelope on the outside. They are obligate intracellular parasites. They cannot replicate outside of a host cell. They are inert outside of a living host. If they are not inside of the appropriate cell, they will not replicate. And virology is the study of those. And lastly, this will be our last slide for today. We'll pick up with the next slide on Thursday. Multicellular animal parasites. And I told you, technically you can see them. The larval stages are microscopic, so that's why we put them in here. So multicellular animals, things like a tapeworm that's shown in the top panel. The bottom panel is a guinea worm. You can see it. It's exactly what it looks like. There is a worm coming out of that person's foot, and they have wrapped it around a stick. That's the way you get rid of the infection. You wrap it around the stick, you turn the stick a little bit every day and start to pull the worm out. If you pull too fast, it's always a girl, will break and either go back up in there or die and cause a really gnarly infection. So very, very gently you pull her out of the wound that she creates to lay her own eggs. You see why I like talking about the parasites. It's a fun section. So we have helminths, which are our parasitic flatworms, and we have our nematodes, which are our roundworms. And of course, the field that studies parasites is parasitology. And that is where we will leave off for today. We will pick up on Thursday with the rest of chapter 1. Maybe we'll make it a little bit into chapter 2, so I'll make sure those slides are available just in case. Otherwise, if you have any questions, feel free to shoot me an email because I have to run to my next meeting. And I will see you all on Thursday.